Okay, it is November 21st. This is ESM 105, and we're going to make some more progress into our pollution and recycling topics, and bioremediation should start today. So let's go ahead and just get back into it. All right. Also, don't forget, of course, discussions. There's one going up today, and your annotated bibliography. Cool. Awesome. So we were talking about recycling. I kind of breezed past this one important word, important term in the recycling industry, and that's contamination. And so I wanted to reiterate that in today's lecture. Contamination is when your material types get mixed together. So if you're filling up a recycling bin, you can contaminate that recycling bin by putting the wrong or non-recyclables into it, right? But also, products can be generated that are already contaminated. So the classic example is the Pringles can, which has at least 4 different types of material in it, and there's not really any way to disassemble it and break it into its constituent materials. Now, recycling goes material by material, so products like this, contaminated products, are especially concerning, right? It's better to have a just plastic cup made of one type of plastic rather than a cup made of paper and plastic. It's better to have a Pringles tube made of just cardboard rather than a Pringles tube made of aluminum, cardboard, and plastic, and then another type of metal usually, okay? So just something to think about if you're considering, hey, how am I going to separate my recycling, or what products am I going to purchase? If you purchase things that are one material type, it's easier for them to be moved back into the production process. And then in recycling, I saw a lot of sources where they were using this phrase of wish cycling, which I just thought was fun. And that's when you just take all your stuff, you think, I think this is probably recyclable, you put it all into the bin, and you're wish cycling. That's fine. We talked about the history of single-use plastics a little bit. We talked about the Keep America Beautiful campaign. We talked about the different members of the Keep America Beautiful campaign and how they focus a lot on consumer responsibility for dealing with waste in public spaces, right? It's not about producing too much waste. It's all about you not letting it just sit there. Sorry, the recorder's in your normal spot there. I'm trying to get the PowerPoint in video there. Okay. Good. So we're going to continue moving forward with our plastic recycling and China, because it's really, really interesting how US recycling shifted in 2017 when China made a decision to stop accepting these MRF residual bales, okay, from the United States. So MRF, what is it? Materials Recovery Facility. What's a materials recovery facility do? It recovers useful materials out of waste, all right? Usually it extracts the high-value recyclables, which can be easily converted into raw material, or at the very least, where the cost of the raw material they generate is greater than the cost of extracting that raw material. So these residual bales, all right, they end up being a huge problem. And sorry for the video there, I wanted to get this in here. These MRF residual bales, okay, they end up being a huge problem because they're everything that can't easily be recycled, all right? So the solution was, for a long time, ship them to an area where it's really cheap to process the MRF bale and extract some value out of them. So for a long time, you shipped these to China. One of the reasons that was practical was because of a trade deficit between China and the United States, where these massive ships were bringing products to the United States ports, but then leaving closer to empty. So the shipping cost becomes pretty negligible, right? You just pop those bales onto the ship and they take them away, okay? So low shipping cost, and a huge issue in recycling and in dealing with waste in general is moving the waste around. That's why aluminum is more recycled than glass, for example. Okay, cool. So importing these MRF residual bales ended up being slightly profitable, very slightly profitable for Chinese markets, as long as you ignored all of the externalities, because these MRF bales tended to be highly contaminated, okay? Meaning, in the recycling terminology, it didn't always include the types of plastic that the MRF bale was supposed to include. So contamination, as I mentioned just a second ago, right, is all about, does this block of recyclables consist of a single materials type? Or at least several materials types that we know we're going to be receiving as the recycling group, okay? So not only was there contamination with different types of materials that they weren't ready to recycle, so everything just ends up going to dumps anyways, there were also poisonous, toxic compounds contaminating these residual bales because of their history of going through the waste processing process. Okay, so in 2017, China makes an announcement that, hey, we're not taking these anymore. They also decided to stop taking a lot of other waste types, and that shifted the landscape of recyclables globally, and especially in the United States, since we've been shipping all of these over there. Now, where do they go? Okay, now where do they go? Well, they're going to places like Malaysia receives a lot of them, but they're starting to crack down on this too, because it's people are realizing that it's not profitable, okay? You have to start dealing with people getting sick and then filling up your own landfills with MRF bale materials that couldn't ever be recycled. So you internalize the externalities and you do the cost benefit, and you find that, oh, there's actually no benefit for us buying this garbage and then just dumping it in our landfills and, paying people to try to sort through it by hand. So the recycling problem of why we don't recycle more usually boils down to an economic discussion of where is the... I've said this plenty of times. You extract the recycled product, you extract the raw material, and can you sell that for as much as you spent in getting the raw material out? If you remember from our ecological economics discussion, there are ways to introduce incentives here. You could have incentive systems for people using recycled materials. And if you require the use of recycled materials, or if you get consumers to prefer to purchase materials that are recycled, then you can shift the market demands and make recycling more feasible, because you can sell the product for more now, so that extra effort you put in to recover the materials is now offset by the sales price. So yeah, there we go. There's some ideas about how could you make recycling work. You decrease the cost of recycling, or you increase the value of recycled materials. All right. E-waste, we talked about e-waste a little bit. We talked about demanufacturing as a process of taking apart a material. All right, it's demanufacturing, right? You manufacture it, you put it together, you demanufacture it, you take it apart. E-waste, all right, big problem because things become obsolete so quickly. Heavy metal contamination and batteries, big issue, okay, with e-wastes. So in Europe, they have this cradle-to-grave philosophy behind tracking their waste, where you have to, if you produce the toxic material, if you produce it, you have to keep track and should be paying within the cost of that product that you are selling. That should internalize all of the costs of transporting the waste eventually, treating the waste, getting it to a secure landfill. All right, and there should be money included there for some office that's supervising this whole process to make sure that you're keeping in line with all of the regulations. So this drives up the cost of producing poisons, putting poisons into the system. Okay, so this cradle-to-grave system is what that cradle-to-cradle book title is really, addressing. It's a follow-up to. It's an elaboration upon it. So we've gone through sort of our hierarchy of waste disposal, right? We've talked about dumps, we've talked about landfills, we've talked about incineration and energy recovery, we've talked about recycling, and then we move up to reuse or upcycling, as it's often called, right? Where you take a product that already was used once and you reuse it. That's reuse. When you're upcycling something, you're taking a product that was already used once and you're using it in producing something else. So upcycling and reuse end up being pretty interchangeable terms, but upcycling, it sounds a little more exciting. Sometimes you'll see like an upcycling art installation every once in a while show up, where someone will use garbage to make some sort of art project, and then that's been upcycled. The thing's not being used for the same purpose anymore, but it didn't have to be, so it was not recycled back into a raw material. You see? All right, great. So other examples of reuse include, hey, wash the glass and plastic bottles and use them again. All right. Focus manufacturing on producing stable materials that can be reused. You make products that are intended for reuse, and it's definitely cutting into your bottom line, because no one's going to pay $300 for your reusable plastic water bottle when you're a factory that focuses on making single-use plastic water bottles. You're probably going to make a lot more money with those single-use products, but market forces and pressures would need to shift, or consumer preferences, How else do we shrink the waste stream? Okay, you produce less waste. Pretty straightforward. Unnecessary packaging, big problem, especially if we're focusing on plastics, okay? Here, increasing the use of photodegradable and biodegradable plastics is a way of shrinking the waste streams. We've already talked about degradation of products, right? Or like composting and things being broken down via decomposition. And put a little bit of nuance on that. It doesn't just break down into dirt, all right? It has to go through this metabolic process. And do we want that? Do we want to be taking all of these high-energy organic compounds out of the system and then breaking them down into low-energy gases that go into the atmosphere? It's a big question, okay? Photodegradable, biodegradable plastics. The idea here is that either light breaks something down or living organisms break something down. But there tend to be a lot of issues with this. For example, if they're just going into a sanitary landfill, they're not going to photodegrade. And the biodegradation rates are really low because it excludes oxygen when you keep layering. You make that delicious pile, it's like a layered cake or whatever, a tiramisu of filth. All right, you don't want that. So it's also a bunch of stuff about when these degradable plastics, what do they break down into? Some of these photodegradable plastics, they just more rapidly break down into like small microplastic fragments. And so, is that what we want? Do we want these technologies? They make the pile go down. But if you want to get really into the weeds on this, the space isn't really the issue, all right? If all of the garbage was clean and free of poisons and didn't contaminate water that flowed through it, then there's space for the garbage. If you look at the actual amount of land area that you need, and think about the fact that you can build a sanitary landfill and then build on top of it, and you could call that carbon sequestration if you want to be a monster, You could totally do that. If you think about that, the real problems come from exposure to poisons in the garbage, and the hazards of workers that have to deal with these waste streams. So, shrinking the waste stream is beneficial. It's a positive thing. It'd be nice to have that happen. Okay, and that's the end of our garbage talk. All right, and now we are into pollution and remediation. So, what is pollution? I know I've had a slide similar to this already, but now I'm reiterating it because it's one of those subjective terms, okay? A lot of these terms that we talk about aren't subjective, but the definition may be misunderstood. Pollution is a subjective term, like calling something a weed. This is always the example that I use, okay? So pollution is something introduced into the environment that can or does cause harm. That's a very broad term, and usually when we think of pollutants, we think of poisons. But other things can be considered pollutants because we introduce other things into the environment, like a bunch of noise, like a bunch of light, all right? That are also pollutants. Noise pollution, air pollution, light pollution. These are all different, types of pollutants. They were introduced into the environment and they cause some harm. The type of harm that they cause might be different. I mean, you think about noise pollution from highways, all right? If you look at animal populations that are within, the number's crazy, like within 10 miles or something like that of highways, they have increased levels of stress hormones, and it leads to reduced life expectancy. So they did a cool study with deer, and they were like, the hazards of highways. Is it just deer getting hit by the highways? And no, it also has a lot to do with the racket. Like all of that noise, it inhibits, the lifespan. It reduces the lifespan of these organisms. So it's another type of pollution. It does cause harm. Okay? Good. Legacy pollutants are pollutants that have a high, what was the word we used? Was it resiliency? No. It was perseverance? No. It was, I have to double back. I want to use my right language. It was, oh my God. Endurance, persistence. There it is. It was persistence. Legacy pollutants have a high persistence. They last for a very long time. Yeah, they're disciplined pollutants. Yeah. Legacy pollutants. It's a term that you'll hear. Where was I? Oh my gosh. Come on, John. Yeah, hey, what's up? I'll get there. I'll figure it out. Glass and plastic bottles, pollution, remediation. Here we go. Remediation. What's remediation? Remediation are efforts involved in removing pollution. So if we understand that pollution's a really broad general term, okay, it causes harm, something introduced that causes harm, remediation can be a broad term, all right? You could have like a light remediation project, light pollution remediation project, because light pollution is an issue and it's reducing quality of life for people in an area or animal life in a given area, right? Something to think about. But we're going to be focusing on remediation of poisons, pollutants, the classic pollutants of poisons in the environment, okay? What can we do? Well, oftentimes pollutants will be immobilized or stabilized. So if there's a pollutant in the environment and your fear is that it's going to spread, hit the groundwater, get into people's drinking water, go into a lake, just spread, go into the atmosphere, get all over the place, you try to immobilize it, all right? So something like vitrification, where you block it off in a big chunk of glass and you're like, cool, now it's not going anywhere. All right. There are other immobilization options that are more focused on chemistry and not being. I'm not a good chemist. I wouldn't even say I'm a passable chemist. But the concept here is pretty mellow. Of just I take the chemical and I put a different chemical on it, and that converts it into a form that's less soluble. All right. Fair enough. Also, we've got removal. Pretty straightforward. You've got a contaminant, you've got a pollutant, you remove it. Get it out of there. Maybe you remove it by physically, they call it dig and dump, is like the term that's used, where you dig up the contaminated soil and you just put it someplace else, usually with a lining or something to prevent water from seeping through and extracting the pollutants. Question? I'm not actually going to talk anymore about vitrification, but it's cool. Beyond the fact that it exists, I don't know the rate at which it is used. Yeah, they encase things in glass. It's usually like liquid wastes that are really, really, really hazardous. So you take the liquid waste, you put it in like a metal container or a plastic container. Let's just assume it's probably going to be a plastic container, right? And then you take that and you encase that in glass. And now it can't leak out unless the glass breaks. Yeah, but it couldn't have happened. Yeah, we put all the toxic sludge in a big plastic bin. It'll be fine forever. Yeah. So glass is less reactive, so it can be more stable if all you're considering is mechanical disturbance or just the effect of the pollutant itself on the container. And that's literally everything I know about vitrification. Yeah. So let's see here. Removal, okay? You can remove stuff doing a dig and dump method where you dig up the contaminated soil and you dump it someplace else. Maybe when you remove it, you put it into a site that has bioremediation going on. So you remediate the soil in some way, and then you bring it back and you put the soil back now that it's no longer contaminated. Or you can remove something with a solvent. So you've got a big plume of toxic waste underneath a community, and so you just pump in, it's called sparging, I believe. You just pump fluid down into the soil, and you include solvents that break down and remove the pollutant, all right, to get it to leave that area. So it's still a removal effort, but it's like a chemical removal, and sometimes the solvent that they use is also a pollutant, but the benefit is that the solvent's more soluble, so it dilutes. And you know the old phrase, right? The solution to pollution is dilution. And you can get a lower acute exposure if you dilute the thing or make it spread out into the environment or go deeper into the soil. Yeah. And the class action lawsuits get moved across, like they get spread out. So each company doesn't go bankrupt. They can just be like, oh, cool. Put them all into one place. Yeah, I'd love that. And remediate the industry. Yeah. No. Wow, good job. I can't believe nobody else ever thought of that. So here we go. Let's keep looking here. I should have hit this button. Here's an example of, this is intended for converting the waste from a solid phase into a liquid phase, how the iron sulfide nanoparticles operate. I wish I could tell you. But we've got some chromium contamination in this area. It comes out of this pollution source. And instead of like letting it just sit here in the saturated zone, you actually sort of break it up, or with the iron sulfide solution, I'm remembering right now, you immobilize it, okay? So this is an example of immobilization, where we've got this pollution source, it's all flowing down into here, and it's just gonna go out and out and out and out, and we don't want that. So you convert it into an immobile form, and now at least the pollutant is stuck here, okay? So there, you throw chemicals in there to do the same principles of immobilizing and removing. Either you solubilize it or you insolubilize it. You can make it become solid, all right? And then it's not gonna move through the water table the same way. Negative impacts of bioaccumulation, okay? So I've got this little 1-2 punch here of negative impacts of bioaccumulation, which we've already sort of spoken about, and then using the principles of bioaccumulation for bioremediation. So if you remember, right, this is a great classic example. Mercury from burning coal, it goes into the ocean. All right, it's in the atmosphere, it settles into the ocean. These bacteria convert it into another form that's gaseous. It accumulates in coastal fog. Lichen, okay, lichens, they bioaccumulate it, all right? And then deer, which eat the lichens, they bioaccumulate it. And so it's now undergoing a process of biomagnification, and now cougars kill and eat the deer, and they bioaccumulate it. And so the methyl mercury just reaches really high levels. And now, in an area that was nowhere near a coal plant, okay, you end up with cougars that have high levels of methyl mercury in their systems. Yes? Yeah, essentially. Yeah. You know, I would bet you dollars to donuts they are, but I'll tell you, when we hit our energy thing, I've got it all laid out, like my raw materials that I'm going to be condensing. And so as I'm hoping that we'll have some good time to talk about coal and the different types of energy that are being produced. But yeah. So this is straight up biomagnification, bioaccumulation, right? But hey, here's the cool thing, right? This is pretty dark, but there's this methyl mercury in the system. It's great, and all the cougars are bioaccumulating it. So if we just take the cougars and we pull them out of the system and we dispose of them safely, maybe we vitrify the cougars, well, then now you've just pulled the methyl mercury out of the system. And that is the idea behind a lot of phytoremediation efforts, okay? So more negative impacts of bioaccumulation. These stories are all over the place. Persistent pollutants that move through the system, they accumulate at higher trophic levels. That's just the principle, okay? Bioaccumulation, biomagnification, but could we use it for remediation? And yes, you can. It's usually done with plants, and it seems a lot less dark when you're using plants, okay? So check this out. Plants have different adaptations for developing tolerances to pollutants. So especially like heavy metal contamination, okay? So check this out. We've got this spectrum here of tolerance. Low tolerance, high tolerance plants, and then accumulation here, high accumulators, low accumulators, all right? So species range in how much they can tolerate these contaminants, these pollutants, and they also range in how much of the pollutant they accumulate. Lettuce is a pretty high accumulation, high tolerance plant, actually. So lettuce grown in contaminated soils can be pretty poisonous, all right? Lettuce is used as a method of phytoremediation, which is plant remediation, where you grow the lettuce on the contaminated soil, and then you take it and you process the lettuce in a plant where the toxic metals can be isolated. Maybe you just throw them in a pile, but either way, you've removed the contaminant from the soil. So the idea here is you use plants that have a high accumulation for the pollutant that you're trying to get rid of, and also a high tolerance for the pollutant that you're trying to get rid of. They call them hyperaccumulators. There are even proof of concept experiments of using these plants for phytomining. And in phytomining, plants, they've done it with gold. So you grow them in a soil where there's gold present, and you find plants that hyperaccumulate the gold. You let them grow, you take them, you process it, and you extract the gold. Now, it's not commercially practical because you can't get enough gold out of the soil to make your money back from the process of extracting all the gold, but that's phytomining, which is pretty interesting. You know, if you get into like some fun, just not really, if you stop letting yourself be bound by what's practical and like what's realistic, you can go into like sci-fi world, and it's pretty fun to think about, oh, all these plants are actually mining all of the things that we need in the environment. That's cool. Humans never have to do it. You just have plants that are engineered to have very deep root systems, and they extract what we need. But for this purpose, for like our what is being done currently purpose, all right, we've got hyperaccumulators as a class of plants that can accumulate high levels of a given pollutant. All right? So, bioremediation. You use living things for remediation. Of course, you're going to understand and have to understand how that species that you're using for bioremediation interacts with its environment. Does it need to be in a very controlled system? Does it compete well enough that if you just seed in the area, you don't need to manage it or remove other weeds or anything like that? So the question is, do we have to manage the site to encourage bioremediators? Now, not all bioremediators are plants. We've got examples like mycoremediation, okay, where fungi are used, okay? And we've got bacterial bioremediation as well. Oh my gosh. Or microbial bioremediation as well, where microbes are used. So I'm just gonna boom, 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 bounce back here real quick. Bioremediation through metabolic breakdown. It's a little bit different than hyperaccumulation for bioremediation, because with the hyperaccumulators, the plant grows and then you take it away, but it still has all the poisons in it, all right? If the bioremediation involves metabolic breakdown, now the soil is above me and this is a root. The root grows down in here and it's covered with a mycorrhizal association, and that's exuding all of these enzymes that are breaking down the contaminant into something less harmful, the pollutant into something less harmful, okay? So now the levels of the pollutant globally are going down instead of you're just relocating the pollutant to another area, all right, and extracting it using plants. So they're both bioremediation, but they're different, right? It's important to recognize the different aspects of bioremediation. Okay, cool. Now, why does bioremediation through metabolic breakdown work well? Okay? Well, some things, like in glyphosate breakdown in the environment, glyphosate breakdown rates are really dependent on the composition of the microbial community, because some microbes can actually break down glyphosate for energy. And if you are a species that breaks down glyphosate for energy, and you're living in an environment with a high glyphosate concentration, then you might become the most abundant type of organism in that environment, all right? So here's the other problem, though. You only have a competitive advantage if glyphosate is present. And if glyphosate's only present part of the year, for example, well, the microbial community is going to shift, and you're not going to be at a high level, when the glyphosate's applied. Question?
There was a person who inoculated some kind of drug, inoculated their stomachs with enzymes that could break down a certain type of plastic. And
Yeah.
the grubs were able to eat plastic and completely bioremediated through metabolic.
Absolutely. Yeah. Oh no, breaking down plastics with bioremediation is probably how we're going to get around all of the plastic in the environment. Burning it works really well. I mean, there are plants that are burning tons of it right now. They're so funky. I'll just go on a slight tirade. If you look up these incineration plants, these waste energy recovery plants, they always have green roofs, and they look really nice. And there's one, I forget where it is in Europe, but the people do skiing on it. It's like an artificial ski slope, and all the time the place is just burning plastic. That's what they do. It's wild, But microbial remediation or bioremediation takes it, breaks it down fully, and as long as the bioremediator isn't a harmful thing, then it feeds into the full ecosystem, and the ecosystem services can kind of take over, which is good. Yeah, absolutely. That's where a lot of my hope comes from, is from bioremediation, which is why I've got so many dang slides about it. Yes, I did watch that. Yeah, it's super fun. In fact, mentioning fantastic fungi seems like a pretty good opportunity to move into mycoremediation. This is the picture. I use it in every single class that I teach, because every single class has a reason to talk about mycorrhizae for a couple of minutes. And there used to be a high resolution version of it out there, and then I lost the PowerPoint that had it, and now it's the only one I can find. But it's such a good picture, okay? What it shows is that, you got these plants. They have a symbiotic relationship with some fungi, okay? And that mycorrhizal associate, effectively it expands the root system of the plant. So it wraps around the roots of the plant and then grows out. It grows into the roots of the plant. There's an exchange of material. And the great thing about fungi is they carry out extracellular digestion. So Terry Hinkle on campus teaches it as like a fungus is like a human turned inside out, all right? Where your stomach is on the outside of your body and you just like lean against a cheeseburger. And he doesn't say cheeseburger, but I'm always thinking about cheeseburgers. You lean against a cheeseburger and the enzymes just come out of your body and then you just absorb the breakdown products. But that's great for bioremediation because you've got this whole network of hyphae growing through the soil and they're all just pumping out these enzymes that are really good at breaking down a wide variety of stuff, okay? And different fungi are good at breaking down different things, and so you know it's pretty good. You select the right mycoremediator, you can get pretty good results, like right here in a fluorine-contaminated soil. I like this example because it hits how do I figure out which mushrooms would be good at this, okay? So here's what they do. They take a soil that was contaminated with fluorine, all right? They cultured a bunch of different strains of fungi with a media made on that soil, okay? And then identified the ones that were good at breaking down fluorine. They add them into a soil slurry, okay? They detect the levels before, they detect the levels after. These ones are good. They put them in to a larger scale soil slurry, all right? And then show that, oh, they removed 90% of fluorine in 288 hours compared to 576 hours without augmentation. Now, this study is pretty controlled. The soil in the system doesn't actually exist as a slurry, like with enough liquid maybe, and there may have been aeration going on, because once again, this is a process of cellular respiration. It's a heterotrophic process, okay, so they need oxygen to be available, but it gives hope and it shows that, hey, these products could work. I'm going to talk to you about bioremediator identification as well and how they try to figure that out just a little bit. So this here has some, I don't know, I put this slide in and I was like, here's a bunch of different mycoremediators that have currently been identified. All right, this table's cool. If you zoom in, you can see like what projects these things can be useful for. They're usually determined by the environment type, the things that they break down. I mean, look, these would be good if there's a sunny, arid environment, okay? So you don't think about fungi in a drier, sunnier environment, but this group might be more effective in that sort of project. This slide's available for you, of course, if you want to look into any of these in more detail. But these are nice, they're well characterized, okay? And that could be a career path that maybe some of you decide to go down. You want to characterize bioremediators? That's great. That's awesome, you know? It'd take, I don't know, just throwing some ideas out there. Okay, that's all we really talk about with mycoremediation. My main key points are like these hyphae, they can go through a large soil area, they expand a lot, and they have extracellular digestion, makes them really high potential. Microbial remediation, huge prospects as well. Now, one of the great things about using bacteria, especially, or like a cyanobacteria or a photosynthetic bacterium or an algae, is they can be genetically engineered pretty effectively. So, for example, you can get photosynthetic bacteria to produce high levels of fats via genetic modification. And then those fats can be extracted as biofuels. And growing those bacteria, all you have to do is put them in a big tank of water with sunlight. And then they just consume carbon dioxide in the atmosphere, and they make a bunch of fats. Then you take them, you process them, you separate out the products, and now you got a biofuel. Pretty solid, okay? Whether or not that genetic modification holds, that's the whole question. Now, don't worry, I'm going to talk to you about options that don't require genetic modification as well, okay? But let's focus, yes, let's start with that focus, okay? Using microbes, single-celled things for bioremediation. You can use indigenous microorganisms, all right? The organisms that are already there in the soil. You can use biostimulation. In biostimulation, you encourage the growth of microbes in the soil. It's usually just aeration, all right? It's usually all the process is really doing. You can use bioaugmentation, where you go and you inoculate with new microbes, all right? That's a little more, people can be more concerned about that, and rightfully so, because now you are adjusting the community composition of the, now you're adjusting community compositions. You're introducing species, essentially, and a history of ecology and a history of human intervention in the environment has made us wary of introducing new species. And then also you can add genetically engineered microorganisms, or GEMs, all right? So these sort of go up in how drastic the human intervention is. Just counting on the microorganisms that already live there, you're just kind of like, cool, keep up the good work, please. All right, so real quickly, let's talk about how you identify a good bioremediator. This figure's wild, but it's because it's from an actual primary literature source, and they're not always great at graphic design, okay? Usually what you do is you take some growth media and you put the main contaminant or the main pollutant that you want to deal with in that growth media. And then you take a bacterium and you put it on that growth medium. And then you let it grow, and then you look at the medium after some time and see how high the levels of the toxic metal in this example are. So any drawdown, any reduction is assumed to be due to the activity of the bioremediator. Fair enough? But it's not just that straightforward always, okay? It's not just that straightforward ever. You want to also know the mode of action, okay? How is the bioremediator surviving, and reducing the concentrations inside of the media, all right? So there's some different primary mechanisms, and one of them, which, as you might immediately identify, is usually a negative thing, is that bioaccumulation. So if the microbe is surviving and reducing the concentration in the surrounding media because it's bioaccumulating it, that's not going anywhere. You're not going to go to that area and then extract all of the microbes. You can't do that. You can't just cut down the field of lettuce. That you can do and pull the lettuce away. But you can't really do that with the bacteria. So just because something shows potential as a bioremediator doesn't necessarily mean that the way that it's doing it is helpful. So bioaccumulation, that's one way that these species can do this. Biosorption is when they just take the poisonous thing and they stick it on their outside. It's like you put it in your pocket so that it can't get into your body. Like, I know I'm going to eat that poisonous Snickers bar. I know it's full of arsenic, but I know it's also a Snickers bar. So I'm just going to put it in my pocket and zip my pocket up, and now it's just stuck on my side and it's not floating around there. Biosorption. It's a perfect analogy, and I won't hear otherwise. And then the good ones are enzymatic action, okay? If it's actually metabolically broken down, now you've got a non-toxic product. Yes, please.
So let's say you have a pile of soil with a bunch of lead or something.
Yeah.
In it, and the lettuce grows upon it, and then the lettuce is not removed, but it just dissolves and
Goes back into the system. Yeah.
and it doesn't. Okay.
Yeah. So it's not really, then if you're not managing it, it's not really bioremediation, right? You're like phytoextraction. Yeah. But it just goes from inside the pile to on top of the pile.
In terms of microplastics, do the synthetic oils eventually, through fungal, bacterial, plantae processes, do they eventually become non-toxic forms if they were to all stay in the pile?
That's a good question of like, can you just put it into a pile and just have it stay there, you know? And that is definitely a solution.
Digested more and more.
Yeah, once again, I hate having to say I'm not an expert in microplastics, but there's a point where the breakdown just gets really low, and the things stop being broken down, and they just start to move through the system and then bioaccumulate. That's a microplastic. Yep. So the hope there is let's get an organism that hyperaccumulates microplastics and then put it into a big bin. Let's put it in a big plastic lined trough. You know, we use macroplastics to solve the microplastics problem. There's not really much of a. It's difficult to think of a microplastic solution. It really is. You're just like, there's a clump. Push the Lego out. Yeah, of your lymph node. Yeah. Oh, I can't wait for that. I'm gonna get all the chips implanted. Yeah, not really. Just a JK. All right. So the key thing I want you to get out of this for an ESM 105, I want you to think about bioremediation via enzymatic action, via actual breakdown. That's better than bioremediation through hyperaccumulation, because then you still have to take the nasty thing and deal with it. But if there's actually an enzymatic pathway that's breaking the product down, well then now you are converting it into a non-harmful form. And that's beneficial. So they consider that when they're identifying bioremediators in studies. It's good stuff, you know. It's very, very difficult to culture the things that are alive in the soil. It's incredibly difficult. If you try to culture, they estimate they can get like 5% of the microbial community can actually be cultured in a lab in a given soil. So there's all these other things that are alive there that we can't study in a lab. So we don't know what they're doing, all right? So what can you do there? You might not know what's happening, but maybe you just use in situ bioremediation with indigenous microbes, where you just say the microbial community will deal with the contaminant, because as the contaminant reaches higher levels, species that can tolerate it will survive more, and species that can break it down for energy will thrive. So instead of trying to control the system, we just increase the growth of microbes that are already there. And this is where we'll start on Monday. I went a minute past, I'm sorry, everybody. This is where we'll start next Monday, and we're just gonna do bioremediation stuff. We're running out of time in the semester. It's a real shame, because I wanted to talk agriculture and water and energy. I'm gonna make some hard decisions over fall break and figure out, maybe we'll do a little water security and then some energy, alternative energy options, and where the energy is generated on the planet. That's where I'm leaning right now. Anyways, have a great day, everybody. Thank you for making it. I'm gonna end this recording. You too.